Hello everyone. In this video I'm going to look at the specific response that we have to infection. This is one of two videos I've done on immunity for ASNA2. Now this one is in comparison to the non-specific response that we have. So on the previous video I made about the non-specific response to infection as part of our kind of immune response, we said that that was immediate and is the same response for all pathogens. Now the thing that defines the specific response are the two things that I've got listed at the top. Now this response is much slower than the non-specific response, but you get long-lasting immunity from it. But it's also specific to each antigen, as the name suggests. So every different antigen has a specific kind of immune response that comes from that. Now the first thing we need to do is define the word antigen, just so that we're all clear on what it is that we're referring to. So an antigen is actually defined as a chemical molecule that stimulates an immune response. So I'll just make a note of that just at the top here. So an antigen, simply a chemical molecule that triggers an immune response. Now this can include surface proteins on pathogens, it can include foreign cells, it could include toxins, damaged or abnormal cells. The immune system recognises these molecules as what are called non-self or foreign, and it produces what are called specific antibodies against them. So we'll just add that detail in there. So when we consider antigens, essentially we're talking about surface proteins. I'll just mention a few of them. So you've got surface proteins. They include foreign cells. They include toxins, abnormal cells. So we'll just put a few down there. So that's the definition of an antigen. Now we've got that definition in place, we can really start to consider this specific response to infection that we have. And the first phase of this specific response we have is what's called cell-mediated immunity. So I'll just put the title there, and this is what we're going to talk about first. Cell-mediated immunity. Now, T cells, a type of white blood cell that we have in the body, they respond to body cells that have been invaded by non-self material. And it's because of something called antigen presentation, which we're going to look at. Now, infected body cells, phagocytes, and even cancer cells can all express antigens from a pathogen on its outer membrane surface. Now, the reason why it's called cell-mediated immunity is because this involves antigens presented on body cells and not merely just antigens within the body fluid. There is something called the humoral response, which we're going to come to in the video, but this is cell-mediated immunity because it's dealing with antigens presented on body cells. So the first thing we need to look at is something called antigen presentation. So let's just draw a big white blood cell here. We've got the nucleus in place. So here we're just going to draw what's called a macrophage. So I'm going to use this in my example. Now, a macrophage is simply a large phagocytic white blood cell. Now, we're going to imagine that that takes in a pathogen by phagocytosis. So we're going to draw the pathogen here as, let's just draw it as this green dot, and it's going to take this pathogen in by phagocytosis. There we go. Now, antigens from the destroyed pathogen get displayed on the surface of the cell, and they're actually bound to a membrane protein, and that protein is known as MHC. It stands for major compatibility complex, and it's actually of the class 2 type. Now that's going way beyond the ASNA2 spec, but we're just going to put that little detail in. So essentially we're saying on the surface, we're going to draw this very simplistically. So on the surface of the cell, we've got a protein called MHC, class 2. We're going to break down that green pathogen. So let's just draw some little blobs here to represent that destroyed pathogen. Now, antigens from that destroyed pathogen are displayed on the surface. So we're going to put a little green dot onto that MHC class 2 protein. Now, all of that process is given a very specific name, and the name for all of that is antigen presentation. So in exam questions, when they refer to antigen presentation, they're referring to what we've just drawn here. So antigen presentation, that's what all of that process is right there. Now, what we need to do is activate what's called a T helper cell. Now, there are blood cells called T helper cells, and we're just going to put here. So these are T helper cells, and they're often abbreviated to T with a very small h. That's the proper way to denote these T helper cells. What they do is have receptors that are complementary in shape that bind to a presented antigen, and it activates the helper T cell, which then will undergo mitosis, so cell division, and produce a whole clone of these T cells, and that has a number of consequences. So this is part 2 of cell-mediated immunity. Essentially, we're going to just draw this in red, for example. In fact, what we'll do is if we were to just zoom in on this MHC class 2 portion here. So we're going to zoom in here. So let's say here's the edge, the surface of our membrane. We're going to draw that MHC class 2 protein. Rather than redraw the whole macrophage, let's put the antigen that's being presented on the surface. Then what you have essentially is this helper T cell come and bind, and you'll find that it has, it's only a very quick sketch. So this is a T helper cell. You find that it has receptors that are complementary to that particular antigen. And what you have to then get is a signal to activate it. So once the T helper cell has bound, there is a signal that activates that T helper cell. So we need a signal to activate. So we've expressed that antigen. A T helper cell with a complementary receptor has bound to that antigen. We then get division, cell division of that T helper cell once it's been activated, and what that does is have a number of consequences. So we're just going to list those consequences here. So firstly, so A, we're going to get increased, we're going to stimulate phagocytosis. So we've got increased phagocytosis. Because if you remember, phagocytosis was our non-specific response to pathogens, illness. So we want more of that. We want more of these phagocytes engulfing these pathogens just to help. What we're going to do is also stimulate, so from the B, production of what are called cytotoxic T cells. And it's denoted with this T with a little letter C, so C production. Now, cytotoxic T cells actually produce a protein called perforin. That's spelled P-E-R-F-O-R-I-N. And what that does is perforate the cell membranes of infected cells, allowing uncontrolled movement of substances in and out. Now, the whole job of the cell membrane is to control what moves in and out of a cell. So if you don't have that element of control, then the cells just simply can't function. And it usually would trigger something called programmed cell death or apoptosis. So we've got these increase in phagocytosis. We've had an increase in these cytotoxic T cells being produced. And we also get production of what's called memory T cells. Now these remain in the blood and tissue fluid, ready to respond to any future infection. So this is the first part of cell-mediated immunity, or rather the specific response. The first part of the specific response having this antigen presentation and then the subsequent cell division of these T helper cells. But what we need to look at now is something called the humoral response, and that involves one other consequence of activating this T helper cell, and that is the B cell activation. So what we're going to do is just shrink the screen, just create a little bit more room. So I'll move this up to the top. So we've had cell-mediated immunity. Now what we need to consider is what's called the humoral response. So let's just consider where this word actually comes from, humoral response. Why is this particular aspect of the topic is named that? It's because many years ago, and basically it's an old medical term, we used to refer to humours as the old fluids that made up the body, like water, fire, for example. This was in a time before actual dissections, anatomical dissections were performed on the bodies. And it's just a term that really stuck. And what it's implying by calling it the humoral response is that this is happening within the kind of fluid, if you like. Now, what we're doing in the second phase of the specific response of producing antibodies that are going to act within the blood. So whereas cell-mediated immunity involved actual antigens presented on body cells, and we're specifically looking at particular cells being presented, or antigens rather being presented on actual surface of cells, here we're talking about antibodies being produced within the fluid of our bodies, within the actual blood. So B cells are another type of white blood cell in the body. Now they have antibodies on their outer surface, and they're already present in the blood. It's a common misconception that B cells suddenly get randomly produced and then they start producing antibodies once a pathogen has invaded the body. These B cells are already there. So these B cells, if we just draw just cell here, and we're going to draw, this isn't particularly to scale well, but we're just going to draw a B cell there with an antibody on its surface. We'll give it the antibody the characteristic sort of Y shape that is seen. So you've got a B cell here with an antibody on its outer surface. Now when the antigen with a specific complementary shape to that antibody is present, the two will attach to one another, and that antigen is taken into the cell via endocytosis. So what we're going to do is imagine, so we had our green big pathogen from up above, and then we broke that down into smaller portions that are acting as antigens, so portions of pathogen that can trigger the immune response. Now what we're simply saying is that, so when an antigen with a specific complementary trait to the antibody binds or is present, the two attach, the antigen is taken into the cell, so very similar to what we had as above, and it's processed on the outer surface again. So this time we'll draw this one in red. So attached to a protein, we're going to present this antigen. So the B cell does carry out antigen presentation, and that's a key thing to note. So we again have antigen presentation with this B cell. Now that's by endocytosis, so it's taken into the cell. But whilst before we had the T helper cell having to have a signal to be activated, we're kind of going to get the reverse here. The B cells will protest the antigen and present them on the outer surface, as we've said. But now these T helper cells, if we just draw a T helper cell again here, these T helper cells will actually bind to the antigen, again, our complementary fit, and what they do is activate the B cell. So I'm not going to draw another picture here. We're essentially just going to do it by arrows. So this T helper cell will complementarily bind to this antigen, and it will send a signal to activate, and it will activate the B cell. Now, once we've activated the B cell, we then get the production of what are called plasma cells and memory cells. So once the B cell this time has been activated, we're going to get the production of plasma cells, but also memory cells. Now, the activated B cell will divide by mitosis to form a clone of plasma B cells. Now, that process is actually called clonal selection. These only survive a few days, but they produce large amounts of antibodies specific, that's the key thing, to the invading pathogen. Now, it's called clonal selection, and if you think about what's happening, you can kind of see where the name comes from. Clonal, because we're getting a clone of these plasma cells. Selection is almost like the body has selected which ones we're needing. If you go right back to the beginning, you have to have that macrophage to present that specific antigen, to have the specific T helper cell bind and be activated, and you needed that T helper cell to be activated to be able to activate this particular B cell. So that's where the name comes from. So we've got clonal selection. That's the process of these plasma cells dividing by mitosis. And it gives rise to what's called, and I'll put it here again, the primary immune response. Now, the memory cells that are produced, they remain after infection and they provide the long-term immunity. They circulate in the blood and the tissue fluid, and they can divide rapidly into plasma cells to produce antibodies when the pathogen is next encountered. And that gives rise to the secondary immune response. So the plasma cells lead to the primary, the memory cells lead or contribute to the secondary immune response. Now, one thing just to quickly finish on, one of the misconceptions that students often have is about antibodies and this idea that they destroy antigens. They do not destroy antigens. Antibodies do not destroy them directly. What they do instead is prepare the antigens for destruction. And there's a number of mechanisms they can do this by. They can coat the virus particles. Should the pathogen be a virus, they can coat that virus particle and prevent it from entering a host cell, because viruses need to reproduce and replicate within a host cell, so they can prevent that. They can clump pathogens, more notably bacterial cells, together, and this is actually called agglutination. And what it does is make it easier for phagocytes to locate, to recognize and actually destroy the pathogens, and they can actually neutralize toxins produced by the bacteria. So you see, antigens don't actually destroy that particular antigen. They can coat the particle, preventing entry into a host cell. They can clump them together, making it easier for phagocytes to essentially do their job, and they can neutralize toxins that pathogen can produce. So there you have it. It's a bit of a longer video, but this is all part of the specific response to disease that we have. It's slow but long-lasting, and it's specific for each antigen. So in a nutshell, you have two main phases: the cell-mediated immunity, where we present that particular antigen, we stimulate T helper cells, and then from that we get an increased rate of phagocytosis, cytotoxic T cell production, and memory T cell production. And then in the humoral response, we use those activated T helper cells to activate specific B cells. And those specific B cells will undergo clonal selection and will form plasma cells, which will go on to secrete specific antibodies, and memory cells that remain in the blood should we get an infection a second time. Okay, hope all that helps.